Scorpions have survived for over 430 million years, evolving venom as a precise biological weapon. Long before venom enters a laboratory, it begins its journey in some of the harshest environments on Earth.
Inside the curved stinger lies a complex venom gland, capable of producing one of the most potent biocompounds known to science.
Not all scorpions are equal. Only specific species produce venom suitable for pharmaceutical research.
Venom evolved for defense and hunting, targeting nervous systems with extreme precision. Gram for gram, scorpion venom can be more valuable than gold because of its medical potential.
What begins in the wild soon enters the world of biotechnology, medicine.
In the next stage, scorpions are no longer wild predators, but the foundation of life-saving medicine.
What looks like a simple droplet is actually a complex chemical arsenal made of hundreds of bioactive molecules.
Scorpion venom is rich in peptides, short protein chains designed to interact directly with human nerve cells. These peptides target ion channels, the microscopic gates that control electrical signals in nerves and muscles. While some venom components are deadly, others can be isolated and transformed into powerful medicines. At the molecular level, even a billionth of a gram can alter cellular behavior. Researchers discovered certain venom peptides can recognize and bind to cancer cells with remarkable selectivity.
Nature spent millions of years perfecting these molecules, far beyond what early chemistry could replicate.
Understanding venom chemistry is the first step. The next challenge is extracting it safely.
To safely harvest venom, scorpions are no longer left to the wild. They are raised inside highly controlled facilities.
Temperature, humidity, and light cycles are precisely regulated to replicate natural conditions.
Each scorpion is housed individually to prevent cannibalism and ensure consistent venom quality.
A protein-rich diet directly affects venom potency and peptide concentration.
Venom composition changes as scorpions mature, making life cycle monitoring critical.
Stress alters venom chemistry. These facilities are designed to keep scorpions calm and stable. Only after months of controlled care are scorpions ready for venom extraction. Venom extraction is performed in sterile labs where precision matters more than force.
Contrary to myths, venom milking is a non-lethal process designed to protect the animal. A controlled microvoltage gently stimulates the venom glands, just enough to trigger release. Each scorpion releases only a microscopic amount of venom, often less than a single drop.
Because of its value, every nanoliter is carefully captured and preserved.
After extraction, scorpions are returned to their enclosures to recover before future cycles.
Once extracted, the clock starts ticking. Venom must be stabilized immediately.
Once venom is collected, degradation begins within minutes. Speed and cleanliness are critical.
Each sample is immediately coded, tracked, and prepared for preservation.
Flash freezing locks venom proteins in their original structure, preserving their medical potential.
Extreme cold halts enzymatic activity that would otherwise destroy these delicate molecules.
Stored at ultra-low temperatures, venom can remain viable for months or even years.
Even microscopic contamination can render venom unusable for medical research.
Raw venom is far too dangerous and unstable for medical use. It must first be purified, molecule by molecule. Even warming venom too quickly can destroy its structure. Every degree is carefully controlled. The first step removes non-functional material while preserving bioactive peptides. Centrifugation uses extreme rotational force to divide venom into distinct chemical layers.
Chromatography allows scientists to isolate individual molecules, some lethal, others life-saving.
Only a small fraction of venom contains compounds suitable for medicine.
Each compound is tested to meet strict medical purity standards.
Once purified, venom is no longer poison. It becomes potential medicine.
Purified venom peptides are now tested for their potential to treat a variety of diseases.
Certain venom peptides can target and destroy cancer cells without affecting surrounding healthy tissue.
By influencing ion channels, venom compounds have shown promise in treating chronic pain and neurological disorders.
Small, controlled doses can transform venom from a deadly toxin into life-saving medicine.
Every step is tested rigorously to confirm that venom-derived drugs behave as intended.
From lab bench to bedside, venom-based medicines undergo stringent clinical trials to ensure safety and effectiveness.
Even with life-saving potential, handling venom requires strict ethics, safety, and legal compliance.
Handling scorpion venom requires the highest level of safety to protect both humans and animals.
Every technician wears specialized gear to prevent accidental stings or contamination.
Scorpions are treated ethically, with careful monitoring to ensure minimal stress during extraction cycles. Strict biosecurity measures ensure venom purity and prevent any accidental exposure to dangerous toxins.
Preparedness for emergencies is critical. Every lab has immediate antidotes and decontamination protocols ready. Ethical review boards oversee every project to ensure humane treatment of scorpions and responsible medical use.
Once safety and ethics are ensured, venom is ready to enter the next stage: medicine production and global distribution. Venom is packaged in specialized vials with tamper-proof seals to ensure safety during storage and transport.
Even the smallest breach can compromise venom integrity, so precision sealing is critical. Each vial is digitally logged for traceability, critical for pharmaceutical compliance.
Venom is stored at sub-zero temperatures to preserve bioactive peptides for medical use.
Venom is transported in highly secure, temperature-monitored containers to maintain stability during transit. From lab to hospitals and research centers globally, cold chain logistics ensure venom reaches its destination intact. Upon arrival, every vial is verified, logged, and stored under strict conditions before clinical or research use. With venom safely delivered, the next frontier is synthetic replication and the future of medicine.
The future of venom-based medicine lies in innovation, synthetic peptides, AI-designed molecules, and lab-grown alternatives. Scientists can now design synthetic venom peptides with precision, reducing reliance on wild or farmed scorpions.
Lab-grown venom offers an ethical, scalable, and sustainable solution for future medicine.
From cancer treatment to neurotherapy, venom-inspired medicine could revolutionize healthcare.
By moving towards synthetic and lab-grown methods, we reduce environmental impact and protect natural scorpion populations.
The ultimate goal is accessible, safe, and effective medicine for patients worldwide.
From a tiny drop of venom in the desert to life-saving therapies in hospitals, science transforms danger into hope.